Thank you so much. What I want though is everyone to leave here today feeling like they at least learned one thing. So please feel free to ask these questions. I understand that this room is about half biologists, half informaticians, maybe have not had experience in biology. I will be going over some biology and I'll try to explain through it, but if you don't understand something, please raise your hand and let me know and I'll explain it. So when we're going to talk about using RNA-seq in cancer research, we've got to obviously talk about cancer. And this is a great figure that was published in Nature a few years ago about how cancer is formed at the genomic level. So if you begin to imagine right here, as we start off as a fertilized egg at the point of conception, if you begin to imagine what it looked like when the first egg was, well, don't imagine what it looked like when you were conceived, but imagine when when that first egg got fertilized, you formed a diploid genome. And as this egg grew into a multicellular organism, as you went from a fertilized egg to gestation to infancy, mutations begin to form in the genome that collected in the cells. First is this sort of intrinsic mutation process. So this is the process as the cells divide. As the machinery that copies the DNA continues to copy the DNA and then be put into daughter cells, mutations occur. These are just natural mutations due to any errors introduced by the polymerases that copy the DNA. Later on, as you're born and you move on to adulthood, you're now exposed to certain environmental and lifestyle exposure. So this could be obviously cigarette smoke, charcoal barbecue, spending too much time in front of a computer screen, whatnot, or cell phone use, as they're saying these days, begin to add additional mutations to these cells that accumulate as your tissues grow. And this is the picture of cells, and these little dots represent certain passenger mutations. But what can happen, though, is in certain adult cells, as the cells begin to accumulate mutations, they begin to accumulate more deleterious mutations. These are represented by these little stars, which we refer to in the oncology world as driver mutations. And these driver mutations begin to transform the cell from what we now have a normal cell to something that has now become more malignant, or what we refer to as cancer cells. Cells that are now rapidly dividing in an unhealthy fashion. So we start off with a normal adult cell, now to what's called early clonal expansion, which eventually becomes a benign tumor. This benign tumor becomes further mutated to become an early invasive cancer, then a late invasive cancer, and then what we don't want, and what unfortunately happens in many cases, is a chemotherapy-resistant cancer. And it's the chemotherapy-resistant cancers that are the ones that grow, metastasize, and then begin to invade other tissues and other organs and eventually kill the patient. When we begin to look at what happens now at the RNA level, so if we have all these mutations occurring at the genomic level, what happens at the RNA level is what we have is a very widespread dysregulation of the RNA that's expressed in these cells. And this is actually a plot from some of my work. This is called a principal component analysis. And each of these little spheres represents a sample. And what this is representing is how different or alike samples are in three-dimensional space based on their gene expression. Here, the samples in red are normal breast tissue. These samples in blue represent triple-negative breast cancers. And what you can see is the normals cluster pretty well together, the triple-negatives somewhat cluster together, but you see this sort of wide separation based on gene expression. And what we begin to realize is that there's dysregulation. Let me get rid of this thing here. That there's a dysregulation of thousands of protein-coding genes in the transformation of normal cells to tumors. Now, to put this in perspective, there's about 20,000 protein-coding genes in the genome. In any given cell, about 10 to 15,000 may be expressed. Here, in the difference between a normal and cancer, 5,000 of those are dysregulated. That means anywhere from a third to half the genome becomes dysregulated as normal cells become tumors. So I think this is really important to think about that these genomic alterations, in essence, cause a wide-scale change in the biology of tumor cells. And what these changes are and what their impact is obviously the focus of a vast amount of research, including ours in our laboratories. So when we take this sort of molecular changes to the human level, obviously the combination of these DNA mutation and RNA dysregulation is lethal. Cancer kills people because of these deleterious mutations and dysregulation. The unfortunate part is that treatment for cancer really hasn't changed much in 30 years. There's been obviously some significant advances in certain areas, but the mainstay, which is chemotherapy, and for those who are not familiar, chemotherapeutics are drugs that target rapidly dividing cells, but they're not specific for cancer cells. They just kill rapidly dividing cells and thus cause tons of side effects. These chemotherapeutics have really remained unchanged. So the most popular chemotherapeutic drug, paclitaxel, first discovered as an extract from the Pacific yew tree. The drug cisplatin, discovered at Michigan State when, by happenstance, a PhD put platinum electrodes into a pool of water and killed E. coli bacteria, and the platinum was leaching off the electrodes. And cyclophosphamide, which actually finds its root in nitrogen mustard, or mustard gas, that was used in World War I and World War II, and then eventually made a chemotherapeutic. The unfortunate part, these are probably 3 of the most widely used chemotherapeutics today, but they were discovered in the 1950s and 60s, and yet they remain mainstay. So while drugs that specifically target cancer cells, so not, drugs that are don't act like chemotherapeutics that are just target all sorts of rapidly dividing non-specific cells, but drugs that specifically target cancer cells are under development, and there have been some great advances, but therapies that drastically extend survival have been few and far between. So I think some major questions in cancer research is, one, what mutated genes or pathways are critical for the development of common cancers? What are the key drug targets for the development of novel therapeutics? What molecular changes cause the chemoresistance that I've mentioned in the first slide? Are there better biomarkers for early detection of cancer? Are there better biomarkers for predicting how the prognosis of these patients and their treatment outcomes? And I guess the most important question, and the reason why I'm here today, is there a technology that can help us answer these questions? Are there technologies that can help us, I would say almost in one foul swoop, begin to learn clues or to answers to these questions and to develop better drugs and biomarkers? And I would say the dawn of the cancer sequencing revolution has really, at least we hope, is beginning to provide answer to these questions. So I think, have you already gone through the history of sequencing by chance? Yeah. Oh, okay. So, all right. So you guys are probably familiar, but is there anyone in this room that's ever sequenced DNA using a gel? Anybody? All right, we got one. We got one. Okay. So you remember how terribly laborious that was, and obviously the advance in technology is what really has spurred on our ability to really sequence and understand tumor genomes, transcriptomes, and methylomes at a much larger scale. This machine here, the XL3730, was the workhorse of the Human Genome Project, and right now the current sequencers, the Illumina, the Solid, and soon the Pacific Biosciences instrument are now the mainstay instruments for the near future. So when it comes to cancer research, this paper here published in Nature in November of 2008 was probably what people will refer to as the start of true whole genome cancer genomics. This was the first ever tumor genome sequence done at the WashU Genome Center. Several of the authors here, and actually you all knows them, Jared, Matt Hickenbotham, are people we've worked with in the past. This was a sequencing of an acute myeloid leukemia genome, the tumor and the normal, and basically it was a proof of concept to show that first whole genome sequencing can be done on cancer tumor tissues, but also it was the first truly comprehensive view of all the somatic mutations that occur in a tumor genome. A very, very little known fact was that this was actually the first female genome sequence also. So it was about the 7th human genome sequence in total and was the first female. So when people have studied cancer genomics, it's a relatively simple strategy. And so this is a strategy minus all the analysis algorithms. But what one does is you sequence the tumor genome of the individual. You sequence their normal genome, usually from their blood. You identify the variants between that are in both of these genomes. You subtract the normal genome variants from the tumor genome variants, and now you're left with their somatic mutations that are caused by their cancer. And what will then be formed in many cases now are what are called Circos plots. Have you learned Circos plots yet? No? Okay. So what this is basically some of the next-gen sequencing scientists were left with a conundrum that said, we've got all this data for every individual. How can we present this data in one figure in a fashion that's understandable, that can be traded among other researchers to better understand their tumor genome? And so they created something called the Circos plot. And the Circos plot here is where the chromosomal karyogram is on the outside circle. So each of this is individual chromosomes. And inside these little rings are the individual elements of the DNA sequencing analysis to explain the different variations that are in the tumor. So for example, the purple line here are intrachromosomal translocation present in the tumor. The green line is intrachromosomal translocations. This blue line here represents variations in copy number in the tumor genome, and these several lines going out represent different sorts of mutations, point mutations, heterozygous, homozygous, large indels, and then small indels out here. But what's really neat, it's a really nice way of taking a single figure to represent an entire cancer genome that could be understood by scientists, and hopefully, I think they want to know is this to be understood by physicians and patients. What was very cool about this sequencing effort in this study is what they did was they then took this small cell lung cancer genome, and based on the average number of cigarettes that this patient specifically smoked, they calculated that there's one mutation in the lung genome for every 15 cigarettes smoked. So basically, that's a lot, I think. I don't know how many cigarettes are in a pack, but I'm guessing you get one mutation for every pack of cigarettes. So not very good. Not very good. So.
And actually, there is a software about this type of figure. If we're interested, we can pass around that information.
Are there any questions, by the way? Yeah, that's a great question. I think it would be probably one SMAC mutation in the cell, in the epithelial cell of the lung for every 15 cigarettes. It's not across every cell in the lung. That's a great question. All right. But I think there's some limitations to doing cancer whole genome sequencing. First of all, it's expensive. This cost here about $10,000 per genome. This is the rough cost I just got from Howard Edinburgh a few weeks ago. Is that pretty much the case? It's pretty accurate. And if you imagine you got the tumor and the normal, so that's about $20,000 per patient. And that does not include the cost of analysis. So you can imagine that the cost now becomes exponentially higher to do whole genome sequencing. The second part with whole genome sequencing is that in some cases it can help direct administration of a drug, but in most cases it does not. So what does this mean? So there's this big push right now in the translational oncology world to sequence a whole genome and based off that sequencing determine what drug to give the patient. And the premise is if a certain drug target is mutated, you then give the drug against that mutated gene, or at least in its pathway. Talking to a scientist, to one of the scientists from the NCI who's doing this, he admitted it, that there's 1,500 FDA-approved drugs. Many of the genes that are mutated in cancer have no drugs against them. So you can possibly sequence a patient, see their mutated genes, but in reality, you have no drug to give them because there's not a drug against the target. So it's a great idea, but it's not really ready for prime time. And then the third part, I think the third biggest limitation to whole genome cancer sequencing is that it doesn't really give us any insight into how these mutations are affecting the transcriptional biology. You're seeing the mutation in the blueprint, but you're getting no indication of what it's doing to the cell as a whole, to the rest of the biology. So I would argue then, especially if you're money limited, the best bang for your buck in order to get the best idea of what's going on in the cancer cells to study the RNA, or the transcriptome, which is the set of all messages, RNA messages in the cell. And there's some benefits to looking at transcriptome, looking at the transcriptome in cancer. One is by comparing transcriptomes. Let's say you compare the transcriptome between a tumor and normal. One can begin to determine therapeutic targets by looking for overexpressed drug targets. Another benefit is looking for early detection biomarkers. There's a lot of people now using next-gen sequencing on serum in order to find circulating RNAs as biomarkers for early detection of cancer. Another one is the potential to create gene signatures that incorporate the entire transcriptome. So right now, a lot of gene signatures are just made with coding genes. With next-gen sequencing, you can incorporate the use of coding and non-coding RNAs to create your signatures to predict both prognosis, patient prognosis, and also treatment outcome. And also, one of the other benefits of RNA-seq is the ability to detect mutations and fusions in expressed genes to lend clues into disease causation, among many others. And one of the other, I forgot this part, but I think the other positive benefits with doing transcriptome versus whole genome is that your search space, where you're looking in the genome, is limited only to those areas that are transcribed compared to instead of looking at the entire 3.2 billion letter long genome. Any questions? Okay, great. So if the answer lies, the answer to our research questions that I posed to you earlier lies potentially in RNA, the best way to illuminate this answer, in my opinion, is the use of next-generation RNA sequencing. And why I like RNA-seq, and I think you may have learned a lot of this in your previous lecture, is that there's a ton of really interesting analysis endpoints that one can derive from the analysis of this data. Differentially expressed genes, precursor microRNAs and non-coding RNAs, somatic gene fusion, mutation, splicing, novel transcribed regions, viral genes if you believe there's a viral implication, non-coding RNAs, intrinsic subtyping, among other possibilities. I know Yun Long is going to go over several of these, I believe, next week.
And also RNA editing.
And RNA editing, I forgot one. That's why there are many others there. That's called the CMA line. So we've got the many others. So I think what I'd like to do for this next part of this talk is really talk more about designing an oncology-based RNA-seq study. So if by the end of this lecture I get you excited to use RNA-seq in oncology, you will now know how to execute the study. It is my opinion, and this is my opinion, that the most important part of designing RNA-seq studies has nothing to do with the actual sequencing. I think that's a really important part that people forget. The upfront design is what's the most critical portion. And a lot of people will kind of work too fast on upfront design, get done with the sequencing, and then go back and say, oh my, I should have done this. And so I think a lot of time spent on the upfront design is key to your success. These include sample numbers. I'm sure you can ask Yun Long how many people will do a 3-sample experiment and expect to get statistical significance. You can't do that. You got to have proper sample numbers when you do these studies. Biological and technical replicates. According to the recent RNA-seq standards released by the ENCODE consortium, you have to have biological replicates. Any good high-throughput genomic, you have to have biological replicates. If you are in a situation where your experiment doesn't allow you to have too many biological replicates, then you really need to do technical replicates. The statisticians need to get an idea of variance within your studies to be able to do any sort of statistical analysis. So this is very key to a successful study. Tissue selection, I'm going to talk a little bit more about this in the next couple slides, but what disease type you're going to use, is there subtypes of that disease, or you can use frozen tissues versus paraffin-embedded tissues, tumor cellularity, how much tumor is actually present versus normal tissue in your samples, optimal comparators. You'll see a little bit more about this later, but what is your control? This is very important in RNA-seq studies. I can't stress enough an analysis plan, sitting down with people like Yun Long and bioinformaticians to really hash out what your analysis plan is ahead of time. Your translational endpoints, so if you're going to work in oncology, how can we apply this to the patient? And what is your downstream validation strategy? So once you're done with the study, how are you going to validate your findings from your RNA-seq? So I believe, again, in my opinion, that doing RNA-seq in translational oncology is really a team science. You're obviously going to start off with your biologist or geneticist or scientist who's going to pose the scientific question, who's going to direct the study, who's going to also do the downstream biological studies. You're also going to need your sequencing center, which here is Dr. Edenberg and Jeanette, who do a phenomenal job. The next thing you're going to want to get on your team, next person, is a medical oncologist. And I'm biased because my PhD mentor was a medical oncologist, and he's right there, and now my colleague. But I cannot stress the importance of this enough, because medical oncologists are the ones who see the patients, and they can help frame your studies, to frame your experiment to answer relevant clinical questions. And so I provide an example that I've seen way too much often in basic sciences. So my example of a not-so-good experiment is using RNA-seq to compare differences in gene expression between benign breast tumors and malignant breast tumors. And you may say, well, Milan, that's a fantastic idea. Cancer is bad, benign is okay, we compare, we find drug targets, and let's go and publish a paper. Well, if you don't have any understanding of how clinical breast cancer works, you're going to see there's a lot of problems with this. One, there's several types of breast cancer, several subtypes of breast cancer. You're not addressing that question. Benign breast tumors can become cancer, so is that really the optimal comparator? If you catch it off early, a majority of breast cancers are really curable. So do you really need to do RNA-seq studies to find a therapy in a drug that's already in a disease that's already cured by chemotherapy? So this is really a poor question. I think a medical oncologist can look at your question and then reframe it and then come up with what I think is a better experiment. Using RNA-seq to compare differences in gene expression between ER-negative breast cancer, which is a subtype of breast cancer, ER-negative breast cancer patients who develop resistance to standard chemotherapy versus those who do not. So now you're framing a much better question, a clinically relevant question. We have a subset of patients who do bad on chemotherapy, who metastasize and eventually succumb to their disease, and now we want to compare it to people who do not. That, to me, is a much better clinical question, and that's something that a medical oncologist can really do to help frame it. You're going to need surgeons and tissue bank, and/or a tissue bank. Access to patient tissues, and Brian and I can speak to this many times, is probably one of the hardest parts of getting a study. We ourselves right now are working in a rare breast cancer called inflammatory breast cancer. It's taken us 1.5 to 2 years just to get the tissues. even though we've had funding for 2 years to do it. So I think having good access to these clinician folks who can get you these tissues is absolutely pivotal. And obviously you're going to need to get IRB and consent to do any sort of sequencing studying on human patients, human samples. You're going to need to get access to a pathologist. And this is something that's missed out by a lot of people. A lot of people will get a tumor tissue in their lab. It says breast tumor. And you know what? It's not breast tumor. I mean, I've seen this multiple times. And, you know, biologists somehow just, I think, can be very gullible when it comes to getting human tissues. They'll just take what they can get and not realize maybe what they have is not really such a good tissue. So my extreme, my very strong opinion, and I think you should always do this, is to take all tumor tissues should be examined by a pathologist prior to RNA-seq. And why I think this is important is we're actually going to do an experiment here in this classroom. So by show of hands, how many here are biology or biology-minded folks? 1, 2, 3. You are. You are. All of you here are. Okay. All right. I'm presenting to you here 3 tissues. I'll tell you that all of these are from the breast. Okay. One of these is breast cancer. One of these is a pre-breast cancer called a ductal carcinoma in situ. It's not a cancer. It's actually just a benign, at that point, benign tumor. And one is the normal breast. Okay. And by show of hands, if you're a biologist, you know, maybe you know this, maybe you don't. But by show of hands, I want you to tell me which one you think is the cancer. Okay? All right? So how many people think this is the cancer? I don't know yet. How many people believe this is the cancer? Okay. How many people believe this one's the cancer? Okay. How many people believe this one's the cancer? Okay, he said it. That is the cancer. That's right. That's exactly right. This one's the cancer. That experiment would have worked better, too, by the way. All right, so this one's the cancer. Now, you can get a gold star if you answer this next question. Ying Long will buy you a cup of coffee. What? Yeah, that's right. What would be wrong with using this cancer in an RNA-seq study? We're a little bit zoomed in, so it's a little difficult, but does anyone have an idea? Costa. There you go, exactly right. There's a lot of normal tissue surrounding that. So if you were to use this tissue just the way it is for an RNA-seq study, what you're really going to get is a mixed genetic profile. You're going to get the tumor and you're going to get the normal. Now you say, Neelan, my God, that is obvious. We would never use a tissue that has a ton of normal tissue in it. You know, do this study with a bunch of normals. Well, I can tell you right now, I can think of 3 papers right now, Nature, New England Journal of Medicine, and PNAS, of people making broad claims on tumor tissues that now just recently have been rebuffed because it's found out to have tons of normal tissue in it. I think this is a very important process. A lot of people forget this process. Your tumor cellularity is homogeneous, and when you do your tissues, you need to really do a good job of making sure you have a high tumor content. Now, the most important teammate, obviously the most important for this course, is bioinformaticians and biostatisticians. Analysis is pivotal to the success of an RNA-seq project. I think you guys have learned this in your course already. The involvement of bioinformaticians and statisticians at the start, get them involved at the start of your project and not the end of it. That's kind of done often. They're going to help you plan your sample size and biological and technical replicates. They're going to ascertain the proper amount of read depth you're going to need to do your study, help you determine the proper sequencing chemistry and platform to achieve these analysis endpoints. And these are things that are done up front. I'm not talking about the back end. I'm just talking about the front end. And obviously, because of the many ways to look at the data, as I've mentioned before, expression, splicing, et cetera, each analysis endpoint requires detailed expertise. So I came up with this graph that says how a bioinformatician works, that the speed and quality of the analyzed data from an RNA-seq experiment is proportional to the expertise of your bioinformatician. That makes sense, right? But in reality, it's really proportional to how much your bioinformatician likes you. And it's the honest to God truth. So I'm here to teach you how to do a study.
And how many beers.
And how many beers. Right, exactly. So please take these tips to heart, what I'm about to give you. 1, do not ask your bioinformatician for verbiage for your grant the day before it's due. Huge mistake. How many times does that happen?
Every day.
Every day. Do not put 5% effort on your grant for your bioinformatician and expect them to work night and day on your project. Not going to happen. Do buy them gifts. Ying Long likes Apple products. And beer. So iPad 3 and Foster's at the same time. If you want to keep your lung happy, and do keep them happy. I think a happy bioinformatician is good results who keeps a happy PI. So work together with them. They're your friend. They are people too. So let's do that. And obviously, after we've got the experiment properly designed and we've got the experts assembled, you're going to have the execution. And I know Yulong probably has already reviewed a lot of the details of RNA-seq, but I just wanted to give some additional tips to some of the things he had already mentioned. If you're going to do RNA-seq, you're going to extract that RNA, use kits that retain both microRNAs and small non-coding RNAs. Why this is important? You know, right now with current next-gen sequencing chemistries, you either do the micro or you do the full-length transcriptome sequencing. If you ever want to validate microRNAs downstream, you'll need those microRNAs extracted from your original tissues. But I had just recently heard on the grapevine that there's going to be a kit coming out eventually soon that will be able to sequence microRNAs and full-length transcriptomes in a single library prep. So if you're preparing tissues right now for an experiment, it would be best for you to extract both, because if you're going to RNA sequence in the next year or so, you'll probably be able to do that. So you want to keep both of them. Use sequencing chemistries that retain strandedness. The solid platform has always retained its strandedness. Illumina originally didn't. I think now almost everyone is using a directional Illumina protocol. So most people are now using stranded protocol, but if you're ever left with the choice, use a protocol that retains the strand of the genome from which your RNA is being expressed from. This third point, there may be some here who may disagree with this, but I believe you should almost always use paired-end chemistries unless the read length of your actual platform, like IonTOR and the 454, is very long. The paired-end really helps you, at least what I've heard, and Yulong may disagree with me, but I heard it helps with both calling fusions, splicing, certain mutations. Really, the paired-end chemistry is going to help you get those analysis endpoints done quicker. And always, 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 with as much as possible, run all your samples together, meaning you want to batch as little as possible. So to illustrate that point, I'm going to show this example. This is a recent RNA-seq experiment in a cancer cell line. Okay. The samples in red, samples in blue are done in normoxia, meaning under regular high oxygen conditions. These samples in red are done in hypoxia, under low oxygen conditions. Okay, and on this PCA, you see a nice separation of the two oxygen status. Here normoxia, here hypoxia. But you see this really large separation here on the x-axis. Can anybody tell me or take a guess at what this separation is? Long?
The technician fell into sleep. The preparation for the second batch.
Interesting. That's close. That's close. Interestingly, the number one source of variation isn't between this major change in oxygen condition, is that these samples were sequenced in May and this was sequenced in December. It's called batch effect. It's this idea in high-throughput genomics that you can have everything the same, but if you prepare the library in a different room, on a different day, whenever the moon is, whatever phase it is, you get changes in what's called the batches. And thankfully, there are software that help you correct for this batch effect, and when we apply the batch effect correction now, normoxia and hypoxia become the most important discriminator between the samples. All I'm saying is that if you're going to run RNA-seq studies, always do them together. Do your samples together as much as possible.
And if you cannot, sometimes you cannot, and you have to balance design. Don't put the blue in May and the red in December. And then once you compare it, you don't know what is the toxin at. So always do it in balanced design. So you may do some of this, some of that, in December do some of this, some of that. Exactly.
Okay, is there any questions on that part on designing a study? Yes, sir.
I do have a question. So actually, you mentioned that you think that pair ends are the best.
I knew it. I knew it.
You know it's coming. Yeah, I know that I would do that if I have unlimited resource. But if my money is tight, I don't know having enough money to do. I mean, pair ends cost money as well.
Right.
So what do you think is the best balance to whether I want to go to pair end or I want to sequence more samples?
Both.
Yeah.
Well, you have unlimited resources, Yulong. Why are you asking? Personal opinion, increase your sample size. If there's a choice between sample size and paired, increase your sample size.
I completely agree with you. That
yeah.
And you also show that in earlier slides that it depends on the biological question you want to target. Yeah. If you want to find out gene fusion, pair end. No, you got
exactly. That's exactly right. Yeah. It depends on your endpoint. It depends on your endpoint. But if you're doing a standard gene expression experiment and you have to choose between the two, then sample numbers are going to be where you want to be. Do they know? Do they understand what paired end is by chance? Okay.
I hope so.
Do you know what paired?
We talked about that at least.
All right. All right. So what I'm going to go now into is I'm going to show two shortened vignettes of some research that my group has done in RNA-seq and triple-negative breast cancer and also in thymic cancers. It's going to sound slightly like a research seminar, but I put some teaching points through it. And it's being used to show an example of how RNA-seq is done in these tumor tissues and kind of show you what you can get from it and sort of our personal successes and a little bit of how I learned some things the hard way. So I'll kind of go over the experiment. Well, first let's explain what triple-negative breast cancer is. So this is George Sledge. He's the head of our breast program here at the Cancer Center with one of his patients. And every time a breast cancer patient comes into the IU Simon Cancer Center, their tumor is going to have a biopsy taken of it. And that biopsy then is going to be sent to a central lab to have certain what's called immunohistochemical stains done to it in order to understand this, to determine the subtype of that breast cancer. So one of the first stains it's going to get is a stain called for the estrogen receptor, progesterone receptor. So a large percentage of breast cancers overexpress this cell surface protein, which is obviously the receptor for estrogen. And if a patient is positive for the estrogen receptor, they're going to receive standard chemotherapy plus drugs that target the estrogen receptor. So these are very popular drugs that target the estrogen receptor, tamoxifen, fulvestrant, and then aromatase inhibitors targets enzymes that produce endogenous estrogen. They're also going to get stained for another cell surface marker called HER2. And if HER2 is overexpressed or over-amplified, in addition to chemotherapy, they're going to receive they could receive a couple other drugs, specifically the drug Herceptin, which is an antibody against HER2, and lapatinib, which is a small molecule against HER2. But there's this sort of subset of patients here, about 15 to 20% of breast cancers that are negative for both, all three, for ER, PR, and HER2, and we refer to these patients as triple negative. And for triple negative patients, in addition to chemotherapy, there is no targeted therapies to give this patient subset. Well, how is this translated when it comes to survival outcomes? So this is what's called the Kaplan-Meier curve. This is the probability of survival. This is the years after diagnosis of breast cancer. Here are all other breast cancers. Here is your triple negatives, and we can see that triple negatives have much poorer prognosis compared to other breast cancers. And a lot of this has to do with the fact that there is no targeted therapies for this subset of patients. So one of the problems with triple-negative cancer is that our ability to define this cancer is a bad start. So to best illustrate this, this is Brian, again, sitting right here, with one of his patients, Rachel. Here, Rachel is 24 years old. No, Sarah, sorry. And every time Sarah came into the clinic, she made this homemade T-shirt that says, "My oncologist is my homeboy." And obviously, he looks very ghetto there, I think. But anyway, if we look at Sarah and we try to describe her, we can call her blonde hair negative, brown eye negative, and tall negative. But you not learned anything about Sarah. You just learned what she's not. And that's essentially how triple-negative cancer is defined today. We define the cancer by what it's not, ER, PR, HER2 negative, not by what it is. So triple-negative makes about 10 to 15% of all breast cancer with a disproportionately higher mortality. It tends to affect more premenopausal women and women of African descent. Many of you may have heard of the BRCA1 gene, which is a hereditary cancer gene. When mutated, predisposes women to a much higher risk of breast cancer. Triple negatives tend to be BRCA1 BRCA1 mutants tend to be when they get cancer, tend to be triple negative. As I mentioned before, there's a lack or a paucity of effective targeted therapies. And as scientists, where can we look to better define this cancer and also to find new therapeutic targets? So what we did was, in our project, we took 10 triple-negative breast tumors from premenopausal women, 10 normal breast tissues from the Susan G. Komen Tissue Bank at the IU Simon Cancer Center. And if you've been watching the news recently, you've probably seen a lot of news about the Komen Bank because they're tied with the Super Bowl and they're doing all these collection events with the Super Bowl. It is the only normal tissue bank in the world. And here at IU, and it's located here at IU, so we took 10 of their samples. We depleted the samples for ribosomal RNA. I think you've already talked about ribosomal depletion. Yes, you did. Sequenced it on the ABI Solid3, whole transcriptome sequencing. The reads were mapped to the human genome using software called ABI Bioscope 1.2. Bioscope is a software specifically designed for solid color space reads. And then the output was further analyzed both using custom software made here at the CCBB and various third-party softwares from ABI, Partech, and Ingenuity Systems. going back to that point I made about the importance of having a homogeneous sample, half of the class would know that the human breast is comprised mostly of fat and muscle and stromal tissue. The origin of breast cancer is actually the milk ducts, which is these things here in blue. And we wanted to understand what were the transcriptional difference between the breast cancers and normal epithelium, or the normal milk ducts, where the origin of breast cancer is. So to be able to do that, we took these normal breast tissues, we sectioned them in small slices, and put them under a special microscope called a laser capture microdissection scope. And what we do is once we put it under the microscope, we take a laser and circumscribe these cells out of the tissue, and this is what we extract our RNA from in order to get this sort of pure population as a comparator. When we did the sequencing, I thought because we've got some bioinformatic geeks in here, you guys would love to see these numbers. But essentially, we had about 57.3 gigabases of total data with 36 million mapped reads on average per sample, and our mapping rate about 63%. So one of the first things that was interesting when we did this study was we did a PCA analysis of just the normals. So we took the gene expression data from the RNA-seq and did a PCA. And what you can see here is you can see this sort of kind of wide variation in the gene expression among the normal tissues. But because we had really detailed clinical data on these samples, when we looked at a clinical parameter, we saw that we saw a nice decent separation of these samples into these two clinical parameters. So these samples in red represent samples in the follicular phase, which is women, premenopausal women who are on days 1 through 14 of their menstrual cycle. These samples in blue are women in the luteal phase, days 15 through 28 of their menstrual cycle. Don't worry, I'm not saying the joke again. So I have this joke that I've used many, many years that I told myself I wouldn't use again because it's gotten old. I saw Brian there waiting to laugh. Basically, it means this was the first time.
Is gene different?
Yes, there are genes different. Basically, because this is actually the first study to do a whole gene expression profile on the normal breast. As a man, I know the human breast better than women do. So that was a joke. So anyway, we saw this nice separation based on this menstrual phase. The point I want to make for the teaching of this course is I think the accuracy of RNA-seq, which I think Yun Long mentioned in the last lecture, is really powerful, and that this change in phenotype between follicular and luteal in normal breast is actually pretty subtle. When you look at the gene differences, the fold changes are not high. A lot of cases less than twofold. But RNA-seq has that accurate ability to really separate these subtle changes in phenotype. Very powerful. When we look at the difference between tumor and normal, obviously now we have this vast difference in gene expression profile. Here we've got our normals, here we have our tumors, and as I mentioned before, there's roughly over 5,000 genes that are significantly different between tumor and normal. So over a third of the expressed transcriptome in these cells. So one may say, well, you know, Milan, we've had microarrays for 10 years. You know, people have been doing microarrays on breast cancer for many, many years. And it's true. So this is some of the very famous work done in breast cancer using microarrays, where when they took microarrays and applied them to breast cancer tissues, they were able to separate the breast cancers into various subtypes. And they're referred to as the luminal A and luminal B, which tend to be the ER-positive cancers, HER2 cancers, basal-like, which tend to be triple-negative, and then this normal breast. But what we've come to learn is that this normal breast really wasn't truly normal. It was actually breast reduction tissue, and it was comprised mostly of fat. And when you read the papers, they'll tell you that most of the genes that they pick up in this class of tissues was mostly from adipose, or fat tissue. So when we do the comparisons here, when the gene expression comparisons were done between these subtypes, these were essentially tumor versus tumor comparisons. So comparing one type of breast cancer to another type of breast cancer. And that can be very problematic because just because a certain gene is differentially expressed from in one tumor type, subtype, or breast cancer subtype versus another, it doesn't necessarily mean that that gene is important in tumorigenesis. It could be a passenger finding. But when you do a comparison of tumor versus normal, as I mentioned at the start of the lecture, by definition, all differences have the potential, and I stress the potential, though, to be important. So the comparator is very important. And to kind of bring this point home, a lot of people really forget that in RNA-seq, or anytime you do a differential gene expression experiment, your differential expression is always compared to a control, relative to a control. A lot of times people forget that. So if we take this example of gene, like the EGFR, the epidermal growth factor receptor, and we say it is expressed at 20 here in triple negative and maybe 2 or 3 here in ER-positive HER2, you can say, well, conclude that in triple-negative disease, this gene is overexpressed compared to ER-positive. But now if you look at what is expressed in normal, now your whole view changes. It's not the fact that triple-negative is overexpressed in ER or HER2, it's actually EGFR is downregulated in ER-positive compared to normal and triple-negative. So always remember, fold change in differential expression is always relative to a certain control. And you say, okay, that's pretty obvious. Well, how does that play out in the real world? So this is actually a comprehensive table of several targeted therapies tested in breast cancer. Actually, all the targeted therapies, almost all the targeted therapies tested in triple-negative breast cancer. And what you have here is the drugs, the target, the protein target they inhibited, and then I'm going to show you the rationale of using these drugs in triple-negative breast cancer based on prior tumor versus tumor data, what we saw in our RNA-seq studies, the tumor versus normal, and then the outcome of the clinical trial. Okay? So the first drugs here are cetuximab, which is made by Inclone, and gefitinib. Cetuximab is an antibody against EGFR. Gefitinib is a small molecule. We looked at previous tumor versus tumor data. There was EGFR was overexpressed in triple-negative breast cancer compared to other breast cancers. We were shocked to see when we did our studies that we did not find EGFR to be overexpressed compared to normal. Actually, the fold change the fold changes were very similar, p-value not significant, and now 3 clinical trials all negative. When you look at the drugs imatinib and dasatinib, they are small molecules inhibitors of a receptor tyrosine kinase called c-KIT. Also reported, many, if you Google it, to be overexpressed in triple-negative breast cancer. We actually did not find it overexpressed. We actually found it downregulated about 6-fold, and 2 phase 2 clinical trials were both negative. Here we have a drug called aniparib, and there's actually a few others now. Olaparib also, both inhibitors of PARP, which is a DNA repair enzyme. Rationale to be overexpressed in triple-negative, also its role in synthetic lethality and DNA repair. We found it to be overexpressed 4-fold. And 1 phase 2 was positive. A phase 3 showed positive results in the refractory setting. More results are coming out now, but this drug seems to be at least somewhat working in triple-negative disease. So I think the important point is the normal control is vital. Okay? What you compare to can have implications downstream of your drug development. So
I think that those the previous misunderstandings is because of the experiment was not designed.
exactly.
It's not because of the technology.
It's not because of the technology, and
It's a design.
it's a design issue, but it's also an availability issue. The Komen Tissue Bank started collecting core biopsies in 2008, Brian, 7, 8, something like that. So the tissues to actually have the best comparator haven't been available when those studies were originally done. Now they are available, and so there's a lot of work now to encourage breast cancer researchers to use the normal tissues derived from the Komen Tissue Bank for their experiments to have that optimal comparator. Yeah. So, as with any RNA-seq study, though, the validation of your results is absolutely critical. The reason why is a lot of people can maybe try to downplay your results as saying, well, that's just an artifact of your sample size, or you're working with some newfangled technology. How do you know that's actually real? Okay? So we went on to validate this in an additional cohort of 26 frozen triple negatives, and another separate, this was a totally separate set of 10 LCM normal tissues. And we did this then with RNA expression experiments using ABI TACMAN assays. And for those who are not familiar, in gene expression world, qPCR is the gold standard for gene expression. So when you want to validate RNA-seq, you want to use qPCR. Then we followed. And then this is the RNA-seq results. I'm sorry, the ABI qPCR results. And again, we confirmed our findings from next-gen sequencing. EGFR not differentially expressed. KIT, the TMDC here in green, is downregulated compared to the normals, and PARP1 overexpressed. When I presented this data, obviously the first question I got, well, what does it look like at the protein level? So for my thesis defense, I got it done for the protein level, and this is the protein level data by IHC. We had antibodies for EGFR and KIT. There's no good antibody for PARP yet, but EGFR not differentially expressed, p-value insignificant between each other at the protein level, KIT significantly downregulated compared to normal. So both at all 3 levels, RNA, qPCR, and IHC level to confirm those findings. I'm just going to show this real quickly. I cut out a lot of my previous slides in finding drug targets. I only showed this one. One of the other exciting things about EGFR and KIT as drug targets is that they belong to a class of protein called receptor tyrosine kinases. And receptor tyrosine kinases are proteins on the cell surface that initiate large-scale signaling cascades that allow cancer cells to divide and survive. Okay? So what we did in this experiment, or in this analysis, is say, let's take all receptor tyrosine kinases in the genome, find out which ones are expressed in these tissues, and then do a volcano plot. And a volcano plot plots the full change of the difference between triple negative and normal here on the x-axis and the p-value of that difference on the y-axis. And you basically form this V-shape where genes are most significantly downregulated on the left and genes are significantly most upregulated on the right. Interestingly, KIT, which has been tested in clinical trial, was the worst. EGFR is here in this sort of milieu here. But what we had was this sort of class of receptor tyrosine kinases here, this group here, that seemed to be significantly upregulated. Interestingly, our number one hit, PTK7, was just recently published by MD Anderson in the Journal of Cancer Research that when they inhibit PTK7 using siRNAs, it specifically inhibits proliferation of triple-negative cancer cells and not other breast cancer subtypes. So I think it's kind of a proof of concept that this sort of method of using triple-negative versus normal is a really great way to find new drug targets. The other great thing about... Yeah.
So I know that decimal cluster and the PTK7 you just mentioned, there are some we can target. How about other protein? Is that potential drug target?
Yeah. Taiwan is right now in... is it phase 1? Genentech is making a drug against Taiwan. At AACR, they presented their preclinical data, and they were working on a drug to get to phase 1. CSF1R does not have any current drugs in clinical trial, but in vitro does inhibit breast cancer cells. So does FB4 and FB6. Are you familiar with if there's small molecules against any of these other ones? I don't think so.
There are some, I don't know, 4 and 6.
Yeah, I'm not
So against systemic group can...
Yeah, that's exactly right. The other neat thing about doing work with RNA-seq is that you can profile the entire full-length transcriptomes. Because Yunlong is going to go over these sorts of small non-coding RNAs, long and small non-coding RNAs next week, I'm not going to talk about them, but I'm only going to highlight one thing I think is really neat, which is the ability to detect novel transcribed regions. So how many of you, when you were doing your undergraduate training, was learned that most of the DNA in the genome was junk DNA? Anyone heard the junk DNA concept? Come on, raise your hands. All right, good. So what was really neat was this paper that was published in June of 2007, which was really just a landmark paper in Nature, basically told us, you know what, we were totally wrong. There's no such thing as junk DNA anymore. That large swatches of the human genome are actually transcribed, producing a large class of non-coding RNAs. So this led us to take a little inspection of areas of transcription that we're seeing in our RNA-seq study, but where there's no annotated genes. So you can imagine we're looking for areas in the genome where we see peaks of reads mapping, so areas of transcription, but there was no gene whatsoever having been discovered in that region. We set some criterion for the analysis. This was the preliminary criterion. We're going to refine it a little bit later. But 10,000 base pairs away from the nearest ref-seq gene, just in case we were picking up maybe a distant exon. It has to be at least 50 base pairs long, the region, with 20 supporting reads. We then did some further bioinformatic analysis to concatenate that data to form exon boundaries with doing a subsequent differential expression analysis. And what we ended up finding is about 43,000 of these novel transcribed regions. But at first we thought maybe this is sequencing noise. But when we cross-referenced this 43,000 against an EST database called AceView, that runs by NCBI, which is ESTs derived from large-scale cDNA libraries, we found 17,000 of these regions to overlap current databases. So giving us evidence that these novel transcribed regions actually exist. The reason why I think we have significantly more is when these EST libraries were created, they were created using something called poly A selection, where the RNA was derived from the libraries using primers against the poly A tail to get away from the ribosomal RNA. We now know there's a lot of RNAs that are not polyadenylated, and in our chemistry, we used a ribosomal depletion, which got both RNAs that were polyadenylated and non-polyadenylated. I'm thinking, I'm not for sure, but that a large fraction of these are those non-polyadenylated RNAs.
So none of these regions are actually intronic regions?
No.
This is just intergenic?
Intergenic, totally intergenic, yeah. When we did a differential expression, we found 6,000 of these genes to be differentially expressed. And when we looked at the number one novel transcribed region to be differentially expressed between tumor and normal, located at chromosome 15, for those of you familiar with Genome Browser, you can see there's no UCS gene, no ENCODE gene, no non-coding, no transfers, so on and so forth. But what was available was a prediction that a gene should be there. So this is the ensemble gene prediction. This is the AceView EST database, and it's a predictive pseudogene. Now, when pseudogenes, a lot of people don't get excited until recently when a paper came out in Nature to really show that pseudogenes are very important in transcription. They act like a sink to microRNAs. So certain cancers, for example, will upregulate pseudogenes to get them to soak up microRNAs that may be trying to downregulate an oncogene. So there could be some importance here. I don't know yet, but we'll see.
But in the ENCODE project, they didn't see the various level of histone markers in the surrounding regions. So I would imagine that if this is the transcription is related to the promoter region, you probably will see some histone 3K4 markers.
Yeah, you should. Yeah, you should. I have not cross-referenced this data against H3K4. Yeah, but that's a great idea.
Probably those kind of tracks can be put open.
Oh yeah, they could. Yeah. As with any next-gen analysis, you really kind of need like one person dedicated full-time to each analysis.
Probably more.
Probably, there you go. So I think to say this idea of being able to discover novel transcribed regions from this data, my kind of summary point is that there's plenty of undiscovered territory in the cancer transcriptome. I think we've just kind of scratched the surface of really understanding what are all the components of the cancer transcriptome. What was neat then, if we do a PCA analysis based on these novel transcribed regions, here's the normal, here's the tumor, and it seems to segregate the phenotype. But it segregates the phenotype with no protein-coding genes, no non-small coding RNAs, no tRNAs, no nothing. So I think this is quite interesting to see this sort of separation of phenotype just based on these NTRs. I'm going to go quickly through the rest of this. The other nice thing about from RNA-seq is the ability to detect mutations. Triple negative is a great cancer to be able to detect mutations because we know that TNBC tends to be familial, has this early onset of disease, which tend to have more genetic basis to it, and it has a higher prevalence in African Americans, lending more genetic basis to it. We already know that BRCA1-2 mutations tend to be triple negative, again, lending evidence to the role of mutations in this disease. This was really inspired by work published in the New England Journal of Medicine a couple years ago, where people used RNA-seq to discover recurrent mutations in these two ovarian carcinomas. Interestingly, this paper published in New England, the #1 journal in the world, was done on 4 samples, 4 RNA-seq samples. It was absolutely impressive. But what they ended up finding, well, I don't have the data to show, but they ended up finding a recurrent mutation in a gene called FOXL2 that's in 99% of all granulosa cell tumors of the ovary. So we thought, hell, can we do it too? One of the questions with limitations in calling mutations from RNA-seq, so this is the teaching point. If detecting mutations, SNV or indels, is your primary endpoint of your experiment, use whole genome or exome sequencing. I wouldn't use RNA-seq. If you can do both RNA DNA-seq when calling mutations, that's going to be the best if you have the money. That's always a stressful, something like stress. One of the big limitations of RNA-seq is false positive mutations can be introduced through the reverse transcription process. So when you extract the RNA and reverse transcribe to cDNA to do your sequencing, the reverse transcriptase is not highly fidelitous, so it can introduce mutations during that process. The coverage is not uniform in RNA-seq. Obviously, in RNA-seq, you're only picking up the areas that are expressed. You don't have this uniform coverage across the genome. You can only detect mutations that are expressed. So if you have a mutation that's a larger deletion, you're not going to be able to detect a large deletion using RNA-seq. And RNA editing can introduce false positives. But nonetheless, you can do it. So here's an example. So this is an output from the IGV browser. They're familiar with IGV? Yep. So here, we're in this gene, keratin 17. Here we have a T mutation that's different from the reference genome. So we did this, and we called these mutations, and then we put them into a pathway approach. And what we ended up finding out, so each gene that's in red is a gene in which we validated a mutation by PCR and Sanger sequencing. And what we realized, in one of our samples, we had a BRCA mutation, but in several of the other samples, they did not have a mutation in BRCA, but they had mutations in the same pathway as BRCA. So it was kind of this new evidence that we're hoping will be publishing in the near future to show that maybe for the rest of these sporadic triple negatives, these non-genetic triple negatives, a lot of the mutations may not be in the BRCA gene, which is implicated in familial triple negative breast cancer, but for the sporadic, they may be in genes that are involved in the same pathway.
So when you do this, you are looking for, actually, it's germline mutations.
Actually, these are somatic, because
well, this is the tumor samples, but this is not really the somatic mutations that, so you're looking for the families and the justification.
Yeah, I didn't, because for the sake of time, I took out sort of the process, but essentially we called mutations, and then we subtracted any mutation that was in DV-SNP or 1,000 Genomes Project.
That's right.
So we got rid of a lot of these common germline variants. So
but this could still be germline.
It could be, yes, yes,
somatic and
yes.
track that only a small percentage of cells will have the recurrent ones.
No, not necessarily. Sometimes it's 20%, sometimes it's 90% of the tumor will have that somatic mutation. It varies. It varies. Yeah. Oh, and just to show, based on that gene expression platform I showed you, we came out when we did the analysis. It was interesting. We always had this one sample, and for 2 years we never knew why the sample was away until we did was away from the group until we did the mutation analysis, and this one's actually a BRCA1 mutant, separated itself from the rest of the pack. The last analysis we're going to show here with the triple negative is the ability to look for gene fusions. I think Yunlong's going to talk about this next week, so I won't go too much into detail. But gene fusions are essentially when cancers will take 2 genes, usually located on separate chromosomes or sometimes on the same chromosome, and fuse the genes together to form sort of a super oncogenic protein. And we were able to use this technology to detect gene fusions that were expressed in triple-negative breast cancer. So here is the first gene and its exon being fused to the second gene and its exon, and whether that fusion was an inter- and intrachromosomal. And we spent a lot of time on this hoping to find a recurrent gene fusion triple-negative as a potential drug target. We actually did not. Our data seems to support, and actually support data from other groups, that in triple-negative breast cancer there's a ton of gene fusions, but they're more random. They're never the same for each patient, and they're different for each patient. So each kind of patient has their own personal gene fusion. My second example here for RNA-seq is going to be in thymic malignancies. This is actually a pretty short vignette, but I wanted to show it just to illustrate one example. So in this study, we took 13 thymus cancers spread across histological subtypes. Hold on, let me ask first. Does everyone know what the thymus gland is? Okay. When I tell my friends, like people who aren't scientists, they don't know what a thymus is, so I better ask. So a thymus is a gland up here, and it's important for maturation of your immune cells. And it's a very rare cancer. There's only about 500 cases in the U.S. per year, and half the cases in the U.S. per year are seen here at IU because Pat Lair, our cancer center director, is a world expert in thymoma. So he wanted to use this technology in this cancer to see if we can begin to understand its causation and hopefully further drug treatments. So we took 13 thymic cancers spread across subtypes, 3 thymuses from normal and juvenile cases, again sequenced on the solid 3+, mapped reads with ABI bioscope, and then did the software analysis. And I think Yunlong showed this figure at your last lecture, but it's worth mentioning again. When we did the hierarchical clustering of the samples, so here are the samples. This is the different subtypes of thymoma, referred to as A, AB, B3, normal. We saw this beautiful separation of the tumor subtypes, of the thymoma subtypes based on their gene expression. So here are the A's, here are the ABs, here are the B3s, normal. Sorry, I should go back. And then, I'm missing a box. And then the C's and B2. Why this was really interesting, is that it really shows that RNA-seq seems to be an extremely accurate technology. On microarrays, you would never see that sort of separation. I don't think you would. I don't think you can. I think in my experience, but RNA-seq seems to be able to discriminate these phenotypes quite well. And the reason why we were kind of excited about this, you may say, well, Milan, why do you need to do a high expensive RNA-seq experiment if you can determine the subtype by looking under a microscope? Well, the only reason why we are able to do it well here is because we have a very talented pathologist who knows thymoma quite well. But if you can imagine a patient in Colorado who may not have access to that expertise, subtyping of this tumor is very, very difficult. So, and interestingly, 3 out of 13 tumors that we had done were reclassified based on our data, and really their patient chart was changed here in the Clarion IU Health database once we did the RNA-seq. So we are actually right now potentially working on sort of a diagnostic tool to use a qPCR diagnostic tool to better subtype these thymomas. So I think in summary, next-gen sequencing is revolutionizing cancer research through the sequencing of cancer genomes, transcriptomes, and methylomes. As I stressed before, I think a team of cancer researchers is absolutely critical to the success of these studies. You're obviously going to need to get your researchers, your clinicians, and your bioinformaticians on board prior to the execution of your study. Good study design, sample prep, and bioinformatics analysis are critical for the success. And obviously, the beautiful thing about being able to do RNA-seq or any of these next-gen sequencing is the number of endpoints that you can then analyze, specifically, obviously, novel targets, cancer-causing mutations, subtyping, development of circulating biomarkers, among a large list of others, but these are a few of my favorite. Because I showed original data, but also the stress, this is the team that I've been blessed to work with. I mentioned the importance of the team, and this is where I'm at. So let me start off with Brian Schneider, who's my PhD mentor and now my colleague, who's a medical oncologist, but we have more medical oncologists, Brian Schneider, George Sledge, Pat Lair, Ken Kessler, and Maria Storniolo. Bioinformatics, the venerable Yunlong Liu, Marcus Breeze, Jared Glasscock, and then several people from Pi Biosystems who helped us specifically with the fusion analysis. Our surgeons, Sue Clare, Bob Goulet, and Ken Kessler, who helped us get the tissues, and then the tissue bank and the normal tissue bank. Our pathologist, Sunil Babe, where he is here, very, very talented pathologist. And then our many biologists who provide additional assistance, Marchea Ivan, Brad Hancock, Nawal Kassem, and several folks in the Komen Tissue Bank. As you can see, these good, successful studies take a lot of good, talented people working together to the common endpoint, and that's going to be the success of your RNA-seq studies. So that's it. So.
Questions? Well, there are figure out the questions. I have one. So let me ask you one question.
Yes, sir.
If you have unlimited resources, if you can sequencing everything,
Yeah.
what is the experiment you're going to do?
I would sequence everybody.
You're sequence everybody.
You told me I was limited resources.
Yeah.
So you're talking about the ultimate cancer experiment per se?
Yeah.
Yeah, you know, we thought about that. Now, there are major worldwide consortiums right now doing the ultimate experiment, the International Cancer Genome Consortium and the Cancer Genome Atlas of the NCI. And right now, they're charged with sequencing 25,000 tumor genomes, tumor and normal, and their transcriptomes across a variety of the 20 most common cancers in the world. To me, that's the ultimate experiment, being paid by your tax dollars.
Yeah. I don't know where that goes anyways. All right, and a second question is, how do you think that the development of third generation sequencing can potentially improve your research?
In the RNA world? Yeah. Well, first of all, I just got some recent insight from the third geners. I was asking for an update on Visigen and PAC. So it seems like they're having a lot of problems even getting the platforms off the ground. There's like been significant issues. So we'll see how long it will take. The beauty of especially in RNA, of third-gen sequencing technology is that it's amplification free. So you're removing any biases that may be added by the amplification. You also get rid of the you get rid of any PCR-induced mutations that cause false positives in your mutation calling. So I think the implication of third gen is more accurate mutation calling and potentially better differential expression. And faster.
So I'm done with my interrogation. All right.
All right.
So let's thank our